So our body is made up of many different cells, and the way these cells communicate with each other is absolutely essential to overall body function. If cell communication isn't working properly, then we're not working properly. So the way cells communicate is often through what's called cell signaling. What cell signaling is when one cell emits a signal to talk to another cell, which has a receptor. So the signal will be emitted from the first cell, bind to the receptor of the second cell, and from that receptor, there'll be a signaling cascade within the cell that will tell the cell exactly what to do in the process that this signal is regulating. It can tell the cell to divide. It can tell the cell to become a different kind of cell in the developmental sense. So there are different ways that these cell signaling pathways can be not functioning properly. For example, the cell signal maybe can't bind to the receptor, or maybe it can bind to the receptor, but the signaling cascade isn't emitted, and then the cell can't be told what to do. So if the cell doesn't do what it's supposed to do, we can get all kinds of detrimental health defects. We can get tumors or bone disorders, metabolic diseases, neurodegenerative diseases. The list goes on and on. All kinds of things are regulated by cell signaling, and when cell signaling isn't working properly, we can get sick. So one of the co-receptors of cell signaling I want to talk about right now is heparan sulfate proteoglycans. So as a co-receptor, that means that it functions with other cell structures or receptors to receive a signal and to emit a pathway. So HSPGs are relevant in many different pathways that are essential to development, and they also function to have other roles like cell-cell adhesion and different things like that. But I just want to focus on the signaling pathway aspect. So the function of an HSPG is pretty dependent on its structure. So an HSPG is composed of a core protein, which is in black, and then these things called heparan sulfate side chains, which I've drawn in red. And the way these side chains are made is through a very complex process. It's a step-by-step process where an enzyme at each step will make a modification of the product generated from the previous step. So what that means is that each reaction in the synthesis of the heparan sulfate side chains is dependent on the previous reaction before that. For example, if you really want to go brush your teeth, the first thing you got to do is get your toothbrush, and then you can get your toothpaste, and then you can put your toothpaste on your toothbrush, and then you can brush your teeth. If you never got your toothbrush in the first place, you can't brush your teeth. So that's kind of what's happening here. The first enzyme in the biosynthesis of heparan sulfate side chains is called NDST, and NDST is kind of like the getting your toothbrush part of brushing your teeth. If NDST doesn't do its job, the following steps can't happen, and we will get a defective HSPG, which again will lead to defects in signaling pathways and defects of our body function. So what I'm looking at is the NDST enzyme, and there are 4 NDST enzymes in mammals, and there's only one NDST enzyme in my model organism, which is Drosophila, or fruit flies. So the gene that encodes NDST in Drosophila is called sulfateless, and sulfateless mutant flies, so genes or cells that, or excuse me, flies that are missing the sulfateless gene won't create a viable fruit fly. So we want to see if we can get the NDST enzymes encoded in mammals, and we want to see if we can get the genes that encode those enzymes, put them into the fruit fly genome, and see if the fruit fly can be rescued. So what that means is we'll take the gene that encodes NDST1 in mammals, put that gene into the fruit fly genome, and see if a viable fly is produced. That is a fly that doesn't look like it's missing the sulfateless gene. So all kinds of benefits can come out of this research. We can learn more about how the heparan sulfate proteoglycans, how their structure is related to their function. We can learn more about the biosynthesis pathway. We can also better understand how HSPGs are regulated. There are many different practical applications for medicine we can also look at potentially in the future. which would be, like I said earlier, if a receptor isn't functioning properly, we can get diseases. For example, HSPGs are often involved in development of Alzheimer's. If we can find out what is wrong with the structure of an HSPG in Alzheimer's, we can potentially look at HSPGs as therapeutic targets for the disease. Following that, we can look at the enzymes that create the side chains, like MDST. We can also look at those as potential therapeutic targets for many human diseases, first by studying them in fruit flies.